All right, I want to revisit the idea of cell membranes because that's an important concept in biology. So the first thing you need to know is, what are they built out of? Primarily a phospholipid bilayer. They are amphipathic, which means they have different regions, some parts that are hydrophilic and some that are hydrophobic. The center part of the membrane right here, those are the long fatty acid chains. They are hydrophobic. And then these phosphates right here that look like orange balls, they are hydrophilic part of the phospholipid. Now, the proteins that are interspersed, the region of the protein, remember proteins are made out of amino acids. The part that's in there where the fatty acids are, those have amino acids that are going to be, right, hydrophobic. And the region of the protein that is up there by the phosphate groups, that's going to be hydrophilic. Remember, those proteins can be channels, they can be carriers, cell recognition, they can be receptors, enzymatic, and they can be junctions as well. Now, you also have cholesterol molecules, the little green that you can see in there, and those contribute to the fluidity of the membrane, kind of holding the phospholipid bilayer together, and also preventing freezing. And you see that just in animal cells. And then you have these carbohydrate side chains. There's one right there, there. And those carbohydrate side chains, part of the glycocalyx, if that carbohydrate side chain is attached to a protein, it's called a glycoprotein, and you can see that one right there. And if it's attached to a lipid, it's called a glycolipid. Now, how do things get across the cell membrane? Well, one way is if you're not large or charged, you can go right through those phospholipid bilayers. But if you have a charge, then you're going to have to use one of these proteins as a channel or a carrier. Going from a higher concentration to a lower concentration, that's just diffusion. So if you aren't large or charged, you can go right through the phospholipid bilayer, and we're seeing that right here. You can see that it's going right straight through. But if you do have a charge, then you're going to have to use either a channel or a carrier. And this is called facilitated diffusion because the proteins are facilitating the process, because if you're like a chloride ion, you will not be able to move through that region of the fatty acids that are hydrophobic, so you have to use that channel or carrier. Now, if you want to go against the concentration gradient, that is not passive, that's going to be active. It's going to require an ATP. And the one most common, 30% of your ATP, is the sodium-potassium pump, which pumps 3 sodiums out for every 2 potassiums inside. That sets up nerves and muscles. And lastly, bulk transport. This is where you can't go through the phospholipid bilayer or a channel or carrier. You need a bigger way to do it. So that's endocytosis, engulfing either pinocytosis, cell drinking, phagocytosis, cell eating, or receptor-mediated endocytosis or exocytosis. Then it goes out of